No, I'm saying.
You're fine. All right, thank you everybody. This is a packed house for to-be Dr. Servais, Shelby Servais. I'm starting a new tradition. And her family all in the back row, all from Green Bay, California, and Boca Raton. Awesome. Shelby is the one, the only, and the first in my lab to defend. And I want to start a new tradition that was a tradition that my major professor did, which is somewhat of a little bit of a roast, but also something that I've added. So this is Shelby the first week. She dove in, came down from Maryland. She had come to me highly recommended by Emma Rossi Marshall and Heather Bechtold at the Carey Institute of Ecosystem Studies in Millbrook. And her undergraduate research advisor spoke extremely highly of her, and she came down and we put her right to work. So her first chapter, she's standing next to the red mangroves that we sacrificed at the end of her experiment. Shelby is an extremely accomplished scientific outreacher, if that's a term. She has done more scientific outreach than anyone I know, and literally. And I Googled to find out all that she's done, because she's done so much. She's put herself out there, and here's a picture of her with fellow PhD Dr. Ben Wilson. And but the central picture here is really where Shelby shines. She's a bit of a soil whisperer. And I've written a short haiku about Shelby, summarizing somewhat of herself and somewhat of her dissertation. Coastal soil erosion, enter salt and phosphorus. What's up, soil microbes? So, tell us what's up, Dr. Servais.
Thanks, John, for that really sweet introduction. That means a lot to me. I'm really excited to share with you guys the research I've been doing over the last 5 years, looking at how microbial functioning in coastal wetlands changes when it's exposed to environmental subsidies and stressors. So before I get started, I'd like to thank my committee members who are here and participating remotely, Dr. Mills, Dr. Zeglin, Dr. Geiser, and Dr. Jaffe, and my major professor, John Komanowski. And I'd also like to take this time to recognize all the people who contributed to this work, like the Komanowski Lab and our scientific collaborators from the Florida Coastal Everglades Long-Term Ecological Research Network and the South Florida Water Management District, the Everglades Foundation, and the Everglades National Park, in addition to all the technicians and fellow graduate students and summer interns that we've had. So to everyone here and everyone I've listed, thank you. I'm going to start off by explaining how subsidies and stressors can influence ecosystem structure and function. So environmental perturbations, like a subsidy, which is a beneficial input to the ecosystem, such as a limiting nutrient like phosphorus or water, can have a boost to ecosystem function and development, whereas stress like fire or saltwater intrusion might have a suppressing or negative effect on ecosystem function. However, it's important to know that it's not black and white. If nothing is always a subsidy and or always a stressor, there's a balance between the two. So if you excessively expose the environment to phosphorus, the limiting nutrient, it may transition from a subsidy effect to a stress effect. Same with fire, which I just referred to as an example of a stress. If there's just the right amount of fire, it would have beneficial effects on certain plant communities and their development. So I'm going to use a model from Odum's perturbation theory that was developed to explain how, in general, subsidies and stressors interact in the environment. And I'm going to kind of refer to this diagram behind me, where at low concentrations, inputs of a subsidy would have a beneficial enhanced effect on the target ecosystem function. And then as you reach a threshold, additional inputs of that subsidy may have more of a suppressed effect on your target function. Whereas a stressful input might have an immediate negative effect on ecosystem function. But the thing about this hypothesis is that it's situational, and depending on what your target function response is, these lines may change, and what is a stressor may actually be a subsidy. So for my dissertation, I'm here in South Florida, and I decided I was going to focus on the stressors and subsidies that are occurring across the Everglades landscape. And specifically, I'm going to focus on the effects of hurricanes, saltwater intrusion, and phosphorus enrichment. Hurricanes represent a periodic but intense stressful event within the Everglades and other coastal ecosystems. As you can see here from this picture in the Everglades, where before Hurricane Wilma in 2005, we had a full canopy, and then after the hurricane, there was a lot of defoliation within the canopy, exposing the soils to sunlight and higher temperatures. And also, for just a few minutes. So also what we've seen from other studies is that following hurricanes, you can increase soil respiration, which represents a loss of carbon from the system. So instead of the soil in this study containing or holding on to carbon, it would have lost carbon from the system following the storm. And we're particularly concerned with the effects of hurricanes because we know with climate change, they're going to be more frequent and potentially unpredictable in their intensities. Additionally, I'm looking at phosphorus effects because phosphorus is the limiting nutrient in the Everglades, which is upside down estuary. So instead of receiving the majority of its nutrients from the land surface, it comes from the marine end member. And you can see that signature of phosphorus availability in ecosystem productivity, and it's related to hydrology. So in the two major drainages in the Everglades, you can see this effect of phosphorus, where Shark River Slough, which is more connected to Florida Bay, has a productivity wedge as phosphorus comes through surface waters, whereas Taylor Slough, that's a little bit more protected, has a productivity peak in the oligohaline ecotone, where marine water upwells and releases phosphorus, affecting ecosystem productivity. We're particularly concerned with phosphorus because as a limiting nutrient, it can have drastic effects on ecosystem functioning, and there's active work on removing phosphorus from the water reaching the Everglades, and we also know that phosphorus is associated with storm-derived inputs. Saltwater is also a perturbation that's occurring in the Everglades, and in the Everglades it's a freshwater to marine marsh, so there's a gradient from the north to the south where saltwater is actually intruding further into the Everglades because of sea level rise. And what we've observed in the field is that following saltwater intrusion, we're seeing rapid changes in ecosystem structure and function. This is a picture from a brackish marsh that has been exposed to saltwater intrusion and is experiencing this phenomenon of peat collapse where the soil has subsided from the plant. So for my dissertation, I broadly asked what happens to ecosystem function when subsidies and stressors occur in tandem. So what happens when you also have the stress of a hurricane with the subsidy of phosphorus? And what I predicted is that maybe instead of having one of these two extremes of your subsidy effect or your stress effect, your response would fall somewhere in the middle. So I'm going to test the interactions of subsidies and stressors across the Everglades landscape. And to do this, I set up a mix of field, mesocosm, and microcosm experiments, where in Chapter 1 I worked in the coastal mangrove environment in mesocosms where I manipulated hurricanes, the stress of hurricanes, so defoliated mangroves, with the subsidy of added phosphorus. And then for Chapter 2 I moved into the freshwater marsh and looked at the effect of salt exposure in combination with phosphorus exposure. And then for Chapter 3, I did a field experiment where we manipulated pulsed dosing of salinity in both a brackish and a freshwater marsh. And to conclude, in my final chapter, I did cross-gradient experiments within both a brackish and freshwater marsh with multiple levels of salinity crossed with multiple levels of phosphorus. And I broadly said I was looking at how ecosystem function changes in face of these subsidies and stressors. But to tailor my research into one compartment, I decided I was going to focus on the soil component because the soil stores approximately 3 times more carbon than the carbon that's stored in land plants. So it's a particularly important reservoir of carbon across ecosystems, and additionally the majority of biogeochemical cycling occur within the soils. So in Chapter 1, as I mentioned, I studied the interactive effects of defoliation and phosphorus enrichment on biogeochemical cycling in these mangrove mesocosms. So just to reiterate, the effects of hurricanes are typically seen as a stress where following wind damage you have a removal of the canopy as shown here in these pictures. But additionally, within the Everglades, we get this deposit of marine sediment that is rich in phosphorus that can act as a subsidy within the environment. So I asked, how does simultaneous exposure to the subsidy phosphorus and the stress of plant defoliation affect mangrove soil carbon and soil phosphorus? And to do this, I worked with an REU student, Julio Pachon, who is now a graduate student at the University of Florida. And together we designed this mesocosm experiment where we had designated treatments of without phosphorus, so minus P, and added phosphorus plus P, with also defoliated and non-defoliated, so minus leaves plus leaves. And we did this in these red mangrove plant soil cores. And I'm not going to talk about the plant responses today to keep a focus on the soil responses, but what I'll tell you is that the defoliated mangroves regrew all their leaves within the 6-week study. And within these experimental units, we had a soil CO2 efflux collar so we could measure the carbon leaving the system. We also had leaf and root breakdown material so we could measure the change in mass over time. And then we also had embedded within a diffuser that slowly diffused phosphorus over the course of our study. And so thinking about what might happen and how carbon might be lost from the system, I just briefly thought about what would be a pathway of carbon loss. So with defoliation, I expected that the soil respiration would increase as plants and microbes are consuming more carbon as they recover from leaf removal, and that litter breakdown might not change, but the soil phosphorus would decrease as plants remove phosphorus from the soil to regrow leaves, and then that soil carbon might also decrease. Whereas with phosphorus addition, I expected, as has been seen in other studies, that soil respiration might increase when you provide it with the limiting nutrient, and that would also mirror responses of increased litter breakdown. And as we add phosphorus to the soil, I anticipated soil phosphorus would increase, and then in line with increased carbon losses from respiration and breakdown, I expected carbon in the soil to decrease. So I'm going to jump right into the results, and this is a leaf litter breakdown that we measured over the 6-week study, and root litter breakdown that we measured. So leaf is on top and root breakdown is on the bottom. The leaf litter breakdown was unaffected by our treatment, so there was no difference. However, the root litter breakdown was suppressed with defoliation. So in the treatments where we had removed leaves, the breakdown of organic material within the soil was slower. soil CO2 efflux, so the amount of carbon leaving the system, was also suppressed within the leaf removal treatment. So this is just a time series of the soil CO2 efflux for the 6 weeks, and the opened circle and triangle is showing the defoliated treatment, and you'll see that it's consistently lower across the time series compared to the treatments where we had not removed the leaves. So when you remove leaves, there's less carbon exiting the system. What we did notice is when we measured nighttime soil CO2 efflux, that there was a positive relationship between the amount of P that was released and the amount of carbon dioxide that was leaving the system. So this trend only occurred in the nighttime measurement that we did at the end of the experiment. So phosphorus might have some connection to losing carbon from the system. And this is this elemental percentages for carbon and phosphorus. And the main thing to note is that none of it was significantly different, so we didn't see treatment effects on soil carbon or soil phosphorus at any of the 3 depths that we sampled. And to summarize my findings, I'm going to bring back the Odum model and just use it to help explain the results. So what we saw is that there's no difference on soil phosphorus or soil carbon, so it stayed in this yellow, like, no effect zone. Whereas for root breakdown and soil respiration, we saw that the defoliated treatments had suppressed soil respiration and soil breakdown, and that the control and the added phosphorus treatment acted similarly. And we only saw an enhanced effect on soil respiration of added phosphorus in the nighttime sampling. Now, for this experiment, I was kind of curious as to why I did not see a lot of changes within the soil elemental composition and how defoliation was reacting in the soil to cause suppression of carbon losses. And I decided I needed to learn more about what the microbial community was doing within these soils. So I decided to implement extracellular enzyme assays as a tool to assess changes in the soil biogeochemical cycling. And extracellular enzymes are released by the soil microbe into the environment so that they can help break down organic material into easily assimilable subunits that the microbe can then incorporate within its biomass and use. So these are the 4 enzyme categories that I was interested in. I had 2 carbon-acquiring enzymes, cellulobiosidase and glucosidase, that are associated with acquiring carbon so that the microbe can use them for energy and biomass. And also we had phosphatases, so a phosphorus acquiring enzyme, and phosphorus is necessary for nucleic acid and phospholipid production, along with aryl sulfatase, which is also a nutrient acquiring enzyme associated with sulfur, which is used for the production of amino acids, and it's a component of vitamins. And to do this, I decided I was going to follow this fluorometric assay where you load these microplates with a target substrate and inoculate with your soil samples in a buffer, and they fluoresce, and you can measure that fluorescence on a microplate reader and get a sense for the maximum enzyme potential. In Chapter 2, then, using these new techniques, I decided I was going to look at how microbial-mediated soil carbon loss following saltwater intrusion would affect freshwater wetlands, and I did this by elevating salinity and phosphorus. And as I mentioned before, both salinity and phosphorus are important drivers of ecosystem function, and we know from Everglades research that they are linked together. So when we have saltwater intrusion in the Everglades, we also release phosphorus that is bound to the limestone, and that makes it more available to the plant and soil communities. We also know that levels of increasing phosphorus can have drastic changes in ecosystem structure, as has been seen with periphyte and phosphorus additions. So my research question for this chapter is how does phosphorus and salinity interact to affect microbial functioning and overall carbon losses in the freshwater wetlands? And to do this, I set up another mesocosm study where I manipulated levels of salinity. So we had an ambient treatment that had 0 parts per thousand salinity, and our elevated salinity treatment, which was about 9 parts per thousand. And we also had within those added phosphorus treatment, and we added phosphorus at about 2 times ambient load, and then we also had the no phosphorus added. And this picture here is of our setup down in Key Largo, and you can kind of see that these plants here are a little bit bigger than these ones down here, and that's because these ones have added phosphorus and these ones don't. So you can almost observe the effect of phosphorus on the plant community. So for predicting pathways of carbon loss, I expected salinity to increase enzyme activities because within the landscape, we've seen that as saltwater intrudes into a new area, there's rapid soil subsidence, and I believed at that time that it might be due to accelerated enzyme activities and microbial processing rates. So with that, I also expected that microbial biomass carbon would also increase. Following the same lines of reasoning, soil respiration would also increase, and then soil carbon, the net effect would be that it would decrease. And then I thought phosphorus would be a similar effect, where enzyme activities would increase with the limiting nutrient being more available, and then microbial biomass would also increase along with litter breakdown rate and soil respiration. To some, a net effect would be carbon loss from the soil. And jumping into the results, what we found was that there was initial suppression of phosphatase and aryl sulfatase activity with salinity. So I measured activity at 57 days when we had reached the target experimental conditions, and then also after about a year. And what we found is that in phosphatase, which is on top, there was a suppression of that enzyme with added salt in our salt and salt plus P treatment. And we also saw the same response in aryl sulfatase. However, in the sulfur-acquiring enzyme, we also noted that there was a mitigation of suppression, so the salt plus P treatment wasn't as low as the salinity alone treatment. And then after a year, we saw no difference within our phosphatase enzyme activities, but we did see that there was consistent suppression of our sulfur-acquiring enzyme after a year in the more superficial soils. This is now showing our carbon-acquiring enzymes, and we saw a similar effect within the glucosidase enzyme, which is more labile carbon, and it was suppressed at 57 days with salinity. And then there was no difference here in the cellulase enzyme. And then after a year, we saw that both enzymes were suppressed with salinity, and that there was somewhat of a phosphorus mitigation of that suppression for both the carbon-acquiring enzymes. And now for microbial biomass carbon, we saw that it was suppressed with salt in the lower depth of the soil that we measured. So maybe the salt had sunken in lower and was more toxic there to the microbial biomass. And then looking at the processing rate, so litter breakdown, so mass loss over time, we saw that phosphorus increased litter breakdown, but salinity had no effect. And then soil respiration, we saw no effect of our treatments on soil respiration. And then for soil carbon, we noticed that at 57 days and at 1 year, there was no effect of our treatments. But after 2 years of continuing this study, we did see that there was a salt effect on the amount of carbon. So the salt-treated cores had less carbon within both depths of the soil that we measured. So to summarize our finding, using Odum's model again, what we saw with the phosphorus-acquiring enzyme was that there was initial salt suppression, but then long-term there was no effect, and they were acting similar to controls. With aryl sulfatase, there was initial salt suppression and P mitigation, but then long-term both salt and salt plus P treatments were suppressed. And then for our labile carbon-acquiring enzyme, beta-glucosidase, there was an initial salt suppression that was sustained throughout the whole year, and then there was also the phosphorus mitigation for the whole study. And then the more recalcitrant carbon-acquiring enzyme was not affected early on, but after a year had suppression with salinity. And then for our breakdown rate and soil breakdown rate, we saw an effect of phosphorus that enhanced breakdown, and then no effect on soil respiration. And then we did see a change in microbial biomass carbon, so the salt and salt plus P treatments had lower biomass carbon than the fresh plus P, which was a little bit higher than what ambient was, or what our control was. And then soil carbon was unaffected within the first year, but after 2 years we did see an effect of salt, and salt with phosphorus. However, the microbial communities don't appear to be accelerating the loss of carbon, because what we've seen is that they're suppressed in their activity and biomass is suppressed. So this kind of brought on the question as to why are we losing carbon if we're not accelerating microbial processes when we add salt? And I looked into some other studies and saw that this is a nitrogen-limited Louisiana tidal wetland. They saw similar effects of salinity, where it was suppressing enzyme activities or having no effect. And then also when they were adding their limiting nutrient, which is nitrogen, they did see an effect, but only at higher concentrations. But so far, with my research, or with this chapter, we have no evidence that enhanced microbial activity is what is causing carbon losses from these freshwater soils. But phosphorus addition may be explaining some of the loss, because we did see increased litter breakdown rates there. Then moving on, I decided I would go into the field and look at the effects of experimental saltwater pulses. So instead of sustained exposure, which is what I did in chapter 2, I'm now doing pulses in the field in both freshwater and brackish marshes to look at how microbial-mediated organic matter breakdown changes. And this is just some background information as to why we might think that accelerated microbial processing is related to carbon losses from the system. This is a study done in Chesapeake Bay across a transect, a salinity transect, and they found positive relationships with 6 different enzymes with salinity. So as you increase salinity, you found enzyme activities to be higher across all these enzymes. So you don't need to focus on each individual enzyme, but the general trend is what's important. And then there was a negative relationship between salinity and the organic matter content of those soils. So this is some evidence as to why we might think we're going to see accelerated processing rates. And in the Everglades, this is particularly important because we have this organic rich peat soil that we've seen to become degraded after apparent saltwater intrusion. And we're concerned that it's because of accelerated microbial processing, and peat in the Everglades is so important because it gives us our elevation. So this is 2 feet of sea level rise with peat intact, and this is 2 feet of sea level rise if we've observed complete peat collapse. And as you see, it's the amount of land that's now covered by water without peat is much greater than if we are able to maintain peat integrity. So my research question was, how do short, so 1 month of exposure versus long-term 2 years, to pulses of saltwater affect activities in root litter breakdown and root litter breakdown rate in both freshwater and brackish marshes? And to do this, we set up 2 field sites in the Everglades National Park. This is our freshwater site. You can see it's a pretty cohesive marsh. And then here's our brackish site that is already experiencing the symptoms of peat collapse. And within each site, we set up these experimental dosing chambers where we added salt once a month. And then within these chambers, we embedded multiple litter bags that were filled with known mass of litter material and embedded within the soil. And we collected those over time, over the course of 2 years. And so, based off of what we saw in our freshwater mesocosms, I expected that maybe a freshwater site would be more stressed by exposure to salt. So as we saw in chapter 2, enzyme activities would probably decrease, and maybe litter breakdown rate would decrease because there's less active microbial community. And I'm not sure what would happen with soil carbon there, but it might not change. And then enzyme activities at the brackish site, maybe this is where we'll see an enhanced microbial degradation. So enzyme activities in a adapted site might be higher with salt pulses along with litter breakdown rates to ultimately decrease soil carbon. This is just some of the pore water salinity data from the 2 locations between our treatments and our controls. The freshwater site, we on average increased salinity 9 times. And then at the brackish site, which is in red, we increased salinity about 1.5 times after dosing. So we know that we're effectively adding salt to these systems. This is the root litter breakdown. So from the freshwater site, when we embedded these breakdown bags, we represented the plant community that was there, and there were 2 main species at the freshwater site that we were targeting. So we have Cladium and Elaeochris, and what we found is that 30 days, so very short term, breakdown was much faster. So the majority of mass loss in these breakdown bags is occurring within the first 30 days. And there's somewhat of a trend within the Cladium species of litter that adding salt might increase mass loss within those. However, that trend is absent after 2 years. So longer term, that initial enhanced loss is not present. And there's no trend really in the Elaeochris. Then at the brackish site, the main species there was Cladium, so we only looked at root litter from that species, and we did see a significant increase in the amount of mass that had been lost in 30 days. So these only experienced 1 pulsed addition of salt water, but we do see that there's enhanced mass loss early on. However, long-term breakdown was much slower, so the majority of mass loss did occur in the first 30 days. And there was no difference between our 2 treatments. So this long-term increased mass loss is muted after 2 years. These are the enzyme activities at the freshwater site. So I have phosphatase, aryl sulfatase, and the 2 carbon-acquiring enzymes at the bottom. And overall, what we saw is that enzymes varied over time and between species, and they were largely unaffected by our pulsed doses of salt water. We did notice that there were higher enzyme activities after about 1.5 years, but there was no difference across the species and with our treatment. At the brackish site, we saw similar trends where enzyme activities varied over time and were largely unaffected by saltwater addition. And we did see higher activities after about 1 year within the superficial depth of the soil. And we did see this one instance within our more labile carbon-acquiring enzyme, where after on the final sampling collection, it was elevated in the salt treatment. So that might be some evidence how microbial communities might be contributing to peat collapse. It's a lot higher activity in the treated chambers. However, that does not relate to any changes in mass loss, so it's not quite clear what's going on there. And so for soil carbon content, we measured this after 2 years, and the main takeaway from this is that we're not changing the amount of carbon within the soil at the brackish site, and we're only changing carbon at the freshwater site in the superficial soil. And what we saw is that there was a lower amount of carbon within the treatments that have been receiving salt water compared to the controls. So this is somewhat of a confusing factor because we're seeing less carbon in the soil, but we're not seeing accelerated breakdown, and we're not really seeing accelerated enzymatic activities. So we're still losing carbon, but we're not sure how that's happening. And to summarize, again, we really saw that there is an initial effect on root litter breakdown, so there's maybe a leaching or early on an effect of salt, but long term there's no increase or decrease in decomposition. They're the same as the controls at both the freshwater and the brackish site. And then long term we saw some variation and some increase over time, but overall there was not really an effect of adding salt on enzyme activity, and that we did see that one enzyme, beta-glucosidase, that was enhanced on the final collection, but overall we don't see how saltwater is accelerating short-term we're not seeing how long-term microbial processing of organic matter is contributing to peat collapse. There's a disconnect between observing soil subsidence and the processing rates of microbial communities. And one of the things that might be happening is that we're working in a predominantly inundated wetland at both sites. So inundation, so water level, can limit the processing rates of microbial communities. And our brackish site was only dry 18% of the time, and our freshwater site was only dry 5% of the time. And we know from a previous study done in Everglades mangroves that inundation has the potential to suppress enzyme activity. So maybe the water level is protecting the soil from accelerated decomposition in these two sites, and salt has little effect because overall inundation is acting as a control. And then we also have some evidence from another study done in Georgia coastal ecosystem, LTER, that is manipulating salinity, both pulses and presses in the field there in a tidal freshwater marsh. And what they found is a lot of variation over time as well, and that the first exposure to a press and pulses didn't have an effect, but after longer term, they did see that presses suppressed 4 carbon enzyme activities, and there was a little bit of an enhanced effect with pulses. So there's a difference in the balance between presses and pulses, but when they pressed with elevated salinity, so they maintained elevated salinity, they saw lower enzyme activities. Moving on to chapter 4, I wanted to look at how multiple levels of salinity and phosphorus can affect soil microbial responses. And when I did this work, I was really excited and lucky enough to work with 2 undergraduate students who served as summer research experience for undergraduate students. So I really want to shout out Christina Morales, who helped me kick off this study, and she's now a graduate student at University of North Carolina. And then Marco Fernandez, who's actually here now, he's still working in the lab. He was a big help in finishing the study the following summer. So thank you to you guys. It was really great working with you. These response surface design experiments are really helpful in informing predictions because you have multiple levels of salinity crossed with multiple levels of phosphorus, so you can pinpoint locations where there may be a threshold. And as I mentioned for chapter 2, we know that salinity and phosphorus availability are linked, so it's important to consider the interacting effects of the two. And then this is also informed by John Komanowski's work, where they were manipulating 2 nutrients and its effect on litter breakdown in streams to figure out threshold responses. So my research question for this final chapter was how do cross gradients of increasing salinity and phosphorus affect soil microbial functioning in freshwater and brackish soils. And to do this, like I said, I set up this response surface experiment where I had multiple levels of phosphorus from 0 to plus 80 micrograms per liter, crossed with multiple 5 levels of salinity. And our freshwater soils were exposed from 0 to 16 parts per thousand, and our brackish soils were exposed from plus 10 parts per thousand to plus 26 parts per thousand. And we went out to a freshwater location and a brackish location in the Everglades, and we collected soil and then placed the soil within these containers that were mesh so that they could be exposed to our treatment solutions. And my predictions for how enzyme activities and biomass, carbon, and soil respiration might respond within brackish and freshwater soils. I now, from multiple studies, think that salinity would act as a suppressor. Phosphorus might act as a mitigator or an enhancer of enzyme activity, and the combination might result in no change. And then with salinity, we expected maybe suppression within the brackish soils, 2 from our chapter 3. And then phosphorus maybe is what is driving collapse in the brackish marsh, so increase these responses, and then the combination might be no net effect. And then just from chapter 2, I'm concerned with the magnitude of responses. We expect freshwater soils might have a higher magnitude response because they've never been exposed to increases in salinity. They're in more pristine locations, whereas our brackish soils have adapted and been exposed to previous saltwater intrusion, so they might have less of a net response. And getting into the data, this is going to be something I want to walk you guys through. First is these 3D plots. So we have our increasing levels of phosphorus here, going from 0 to 80, and then we have our increasing levels of salinity going from right to left to our response variable, which will be, in this case, phosphatase enzymes. And the important thing to notice is that if there's not a plane, so this one has a plane, if there's not a plane, that means that our treatments were not affecting our response variable. So I'm not going to talk about those graphs, and you don't need to focus on the trend there because there's no significant trend. But when there is a plane, I'll just briefly describe what's going on there. So for our phosphorus-acquiring enzymes, we saw that freshwater activity was decreased as we increased salinity at this 2-week sampling point. And then there was no effect of salinity or phosphorus on our brackish soils for phosphatase activity. Aryl sulfatase, or sulfur-acquiring enzyme, we didn't see an effect of our treatments on the freshwater soils. However, we did see that aryl sulfatase activity in the brackish soils at this 5-week sampling point was related to salinity. So as we increased salinity, we saw higher aryl sulfatase activities too. Soil respiration, I'm skipping the carbon enzymes because we didn't see an effect at any time point or within fresh or brackish soils. So I figured I'd just kind of keep it simple here. So for soil respiration, we saw that freshwater respiration was decreased with salinity, so there was somewhat of a suppressing effect of salinity there. And then our brackish soils were more responsive to added phosphorus, and respiration increased with phosphorus there. But one of the things that I want to point out too is you'll note the difference in the respiration rate. So the freshwater soils have a much higher rate of respiration than the brackish soils here. So they're less active. And then for microbial biomass carbon, we didn't see an effect on our freshwater soils at either time point. And then for brackish soils, we saw that biomass was increased with salinity. So as we raised the salinity, we did have higher biomass in the brackish soils, which might also explain why we saw the higher aryl sulfatase activities. But again, in terms of the magnitude, we saw way less microbial biomass carbon in the brackish soils than we did in the freshwater soils. So even though we saw changes in biomass, it was not the same as the difference between the freshwater and the brackish soils. So to summarize, this one's a little bit different. I just decided to look at the overall difference between our freshwater and brackish soils, and what I saw was that the brackish soils have very low enzyme activities and low respiration and low microbial biomass carbon. So their processing or their function is much lower in general than the freshwater soils, even within and across the treatments. But what I was curious is, like, how do we get from this pristine, cohesive marsh to this degraded, pockmarked ecosystem where we're seeing peat collapse if there's not a difference in or accelerated microbial processing rates? Like, if this is going from here to here, from 40% carbon to only 11% carbon in these soils, why is that happening if the microbes aren't accelerating their processing rates? And fortunately, I was lucky enough to be doing these experiments at the same time when other graduate students were also manipulating or working with me and looking at different effects. So this is some of Ben Wilson's data from his dissertation, and what I found is that you've got to look at the bigger picture. If it's not the microbes and peat is collapsing, what is going on here? And what we saw with his data is that it's changing the below ground plant biomass. So the inputs of carbon, which is what the microbial community is processing, are much lower when we add salt. So this is our freshwater site in the field, and we saw a significant difference between the amount of plant biomass, or below ground plant biomass, so roots, was much lower with added salinity. At the brackish site, we also saw the same effect. So as we elevated salinity, it's much lower. There's much lower plant biomass, so fewer inputs into the soil. And also the difference between the freshwater control and the brackish control is significant. So even the brackish site in general just had less below ground plant biomass. Additionally, with our salinity and phosphorus experiment, we saw that within the salt added, so this 9 parts per thousand treatment cores, we saw suppressed or lower below ground biomass. Now, if you remember from that experiment, we saw accelerated decomposition with added phosphorus, but not really a change in carbon. And that's probably because phosphorus enhanced below ground inputs in those plants. So the balance between what's being produced below ground by the plant, so the inputs, is disconnected from the processing rates of the microbial community. The processing rates don't really change or they're suppressed, but the amount of inputs from the plant are significantly lower, so there's more loss of carbon because they're disconnected. So my major conclusion from these saltwater intrusion experiments is that in order to understand the net ecosystem response, it's necessary to study the balance between the inputs and the microbial processing rates. So if I just did my own research, I would have found that salt is a stressor, processing is slowed with salt. But if I combine that with the decreased inputs from plants, then you start to see how peat might be collapsing in these ecosystems. And this kind of makes sense because within the microbial community and within the genetic codes there, these biogeochemical cycles, so the enzymes that are being produced, are fairly functionally redundant. You can have the community composition change, but the net function might stay the same. As opposed to plants, which don't transition into new communities as fast as the soil microbes, and they're pretty much stuck there, so they change their function as opposed to their composition at different time rates. And then to summarize across the ecosystem how different subsidies and stressors are interacting in the landscape, in the coastal mangroves, we saw that a disturbance that's administered to the canopy of the plant community can have below ground consequences for carbon storage. So we defoliated the mangroves and we saw effects of that defoliation in the below ground compartment. In our freshwater marsh, we saw that salt can decrease the functioning of the microbial community and that phosphorus can sometimes mitigate that effect. But overall, we didn't see lines of evidence that it was the microbes leading to peat collapse. And then across the brackish and freshwater marshes, we saw that long-term pulses of salt did not change the processing rates of organic matter at either site. And then legacies of previous exposure to saltwater intrusion might dictate the responses to salinity and phosphorus gradients. We saw that there was an inherent difference in the processing rates of the brackish soil compared to the freshwater soil that indicate carbon losses, but not necessarily directly related to what the microbes are doing. And so with that, I'll take questions now.
I'm just going to ask that the committee hold their questions until the end, so this is open questions. And there's a microphone in my mouth and also one that Matt has, and if you use that one, you have to hold it down. But if you use this one, you don't.
Hi Shelby, thanks, that was great. I learned a lot. But I have a question about chapter 1, and the question is, I like that you were looking at how stressors and subsidies could combine. What happens to a system if the subsidy comes at the wrong time, right? So you lose your leaves and you get this subsidy, but you've got no machinery to use that subsidy. Does that explain any of the kinds of things you're seeing out there?
That's a really good point. That's a really good point, thank you. So the timing of the exposure, so when we defoliated those mangroves, it occurred at the very beginning of the experiment, and we administered the phosphorus at that same time. However, we know from other studies from mangroves that the timing of phosphorus exposure does affect the response. So if we had previously loaded those mangroves with phosphorus, we would have saw that there could have been more of a negative effect with added phosphorus, because phosphorus can increase the amount of above ground biomass and make a storm even more dangerous for those communities. Also, the plant biomass, if it's enriched with phosphorus, it might decompose faster. So the biomass that we used to measure litter breakdown wasn't enriched with phosphorus, but if it had been from previous legacies of exposure, we might have seen maybe more carbon loss. So that's a good point. It's hard to know how to generalize because the timing and the duration and the relative timing is important. Yeah. Yes, Steve.
So on your soil respiration collars, do those include roots?
Yes, so the respiration collars included roots, but that was mostly chapter 1. But later on, I did soil incubations of just soil, so they did not include roots.
So in chapter 1, I'm just wondering. So you saw that big decrease with defoliation. You'd expect that since there's much less carbon to be able to supply the roots for root respiration.
Yeah.
So do you have any idea how much of that change is root respiration versus changes in soil respiration?
No, and I think that's a really good point that we weren't considering at that time, but in future experiments, when I was measuring soil respiration, it was on removed soil, and we also have the collar with roots from Ben's measurements, so we can kind of match those two together in the later chapters, so we can maybe distinguish between the two.
One more question.
Okay.
So when I think about plants and the effects of salinity, I think about two things. There's the osmotic effect, and then there's the effect of the ions, so the toxicity of sodium ions or chloride ions. Do you have any sense of whether the osmotic effect is important here or not?
Yeah, so that's one of the reasons why we might see suppressed enzyme activities is producing an enzyme is energetically expensive for the microbe, so that they also have to produce osmolytes that help maintain cell turgor. They might transition between the two, but salt could also act as like a killer or suppressor of the total biomass of the microbes, as we saw in chapter 2. Also, there's an interaction of salt in the extracellular environment that might affect the enzyme activities too. So they might act directly upon the enzyme as it's in the environment, and they might also directly affect the production within the cell. Does that answer your question? Yeah.
Great job, Shelby.
Thanks.
You can hold it.
Okay. We just had a really large experimental defoliation take place. And have you thought about sort of in some of the areas where the soils are more frequently and well flushed, where that litter was carried offshore, versus some of those areas that aren't as well flushed, where that litter might still be in place, and think about the results of your experiment and how you could potentially scale that up? Or any thoughts about that?
No, I haven't thought about that. So the accumulation of biomass that had been removed and deposited elsewhere.
Well, having these defoliated forests, some with standing litter,
Yeah.
others without standing litter.
Yeah, and I think too, like the litter would be standing on the surface. So if it had been deposited somewhere with an intact canopy, it would have different effects than if it was deposited directly beneath, where the canopy was allowing a lot of light to penetrate. I had not really thought about how that might affect it, but if the leaves were removed from a different area that maybe had been phosphorus enriched or not, that might affect the processing of it there. And yeah, is that?
Well, I was thinking in terms of soil respiration and the suppression that you saw in
Yeah,
your
so there might be
versus-
some, from mine, there might be some lines to think that you don't necessarily get this huge breakdown of that organic matter following the storm. There's something going on where in the defoliated area, the respiration within the soil, which might be plant root related, is lower. So there might be a buffer of carbon loss following a storm. So we lost the leaves, but the breakdown of those leaves is a lot slower.
Hey, Shelby. Hey, great stuff. It's really exciting to see all the work come together. So now that you had time to synthesize all this and reflect on all of the measurements you've taken, and you saw a lot of, no responses to some of these subsidies and stressors. So, in the future, if you could take another measurement or do a different experiment, what would you do to try and better understand how microbes respond to subsidies and stressors? And what would that tell us?
That's a big question. I could basically do another dissertation on. So if anybody wants to fund that, no. So there are a few things. I think one of the things that I really am curious about is the direct cause of the changes in the enzyme activities. And is it externally or internally in terms of is it the microbes' production, or is it something else going on in the environment? There's ways you can test that with enzyme addition, so you can add enzymes and see the change. That would be really cool. There's more molecular approaches that you could do, and I actually have archived all the samples that I've ran enzyme assays on. We have a paired sample that's been archived in a -80, and there's a few people that I've been interacting with, and some of Dr. Mill's students may go on to process those samples and figure out more about the genetic code of the microbial community. So that would be interesting. And then also looking at maybe the effects of drought too, because that soil submergence might be acting as a latch.
Thanks.
Good job, Shelby. Very good presentation. And so, how much did other people increase salt concentration in other wetlands to see the effects that is shown, and what's the effect of the composition of bacteria on your results?
Yeah, so from the 3 studies I showed, the Louisiana nitrogen-limited wetland, I think it was about from 0 to 3.5 parts per thousand change in salinity, and then from the Georgia coastal, I'm not quite sure the change in salinity, but the Marissi and colleagues paper that did the transect went from 0 to 7.5, and that was across a natural tidal wetland. I think that the location of these marshes is really important to consider, because the Everglades is highly organic matter, and it might be experiencing the effects of sea level rise differently than these tidal freshwater marshes. So it's really awesome that my research fits into this more eastern coast studies of saltwater intrusion. Sonia?
Good job, Shelby. I don't usually work that far up in the marsh, so I don't know if there are issues with co-limitation with nitrogen, and if you had added nitrogen instead of just phosphorus, what do you think would have happened?
Yeah, that's a good point. I'm not sure what would happen, and I don't know if we reached nitrogen limitation in our study. These soils are really phosphorus limited, and what we've seen is the plants kind of suck up the added phosphorus really quickly, so it's hard to detect its presence within the soil. So I honestly am not sure we even reduce the phosphorus limitation as much.
Hi, Shelby. Again, I have a million questions, but I promise this will be my last one. So when you guys have seen this heat collapse, right? And you created cores of sawgrass that you put in there to do your experiments with, right? It was a fixed size. Has anybody looked at the size of those plugs to see if heat collapse happens quicker in a small plug compared to a big plug? Like if you have a big chunk of it, right, my head says it should take longer for that heat to collapse, right, than a smaller piece of it. Has anybody looked at that?
So I think we have evidence that we could answer your question. In terms of physical peat collapse in the field, we don't have the exact data we need for that. We set out sediment elevation tables now, so in the future we'll be able to physically measure heat collapsing there. But in the mesocosm experiments, Sean Charles did measure elevation change, and what he saw is that salt is decreasing the elevation, and well, I didn't show data from this other experiment that had smaller cores, but we do have different experiments where some of the cores are big and some are small, and I'm not sure if we're really seeing a difference between the rates of that, but we are able to measure in both situations it collapsing.
Thank you.
Yeah, we saw the exact same elevation loss, whether it was a smaller one or a bigger one.
Okay. Oh, that's interesting.
But relative to the ecosystem, they're still both really small plugs.
Yeah.
Right.
Any other questions for Shelby? Okay,
Yay.
good job. I just want to give one last shout out. Shelby actually accepted a fellowship through the National Academies of Sciences that I didn't mention, as did Ben Wilson. So these two are going to be like plants and soil like forever.